I'm going to start recording. Okay, so let's take a quick look at these results. So as to the question, do you use internal standards in a typical targeted metabolomics experiment? If so, how many internal standards do you use? The most common answer was 6 to 10, with second place being 11 to 20. So it looks like we've captured a moderate range. There were a number of responses that said 1 as an answer, which I expected to see. Relatively few use over 20 or more than, or even more than 40 internal standards. As to question 2, for an untargeted metabolomics experiment, the most common response was 2 to 5. So the number of, that's interesting, not unexpected, but the number of internal standards in an untargeted metabolomics experiment actually works out to be lower than in typical targeted metabolomics experiment. It makes some sense because internal standards are really widely used in targeted metabolomics. They're a little bit more variable in untargeted, but there were responses across the range. Question number 3, what do you currently use internal standards for? We got a good number of responses for all of the potential answers. The most common response was quality assurance, quality control to assess a stability of retention times. Looks like the next most common was a two-way tie between absolute quantitation and assessing chromatographic performance. But all of the responses aside from other got a fairly substantial response to that. When selecting isotopically labeled, question 4, when selecting isotopically labeled internal standards, which isotope do you most commonly use? Deuterium, carbon-13, or some of both? This is an interesting question, and I've seen various results to this survey, and I think we've more or less reproduced those results here today. So the majority of responses at 43% said primarily carbon-13, but deuterium when necessary. Only 9% said exclusively carbon-13. Only 7% primarily deuterium carbon-13 when necessary, and 7% exclusively deuterium. And then a substantial number, 15% said mostly use non-isotopically labeled internal standards, which are another valid type of internal standard. What is the most important factor in your selection of a specific internal standard? It was a two-way tie between isotopic purity and availability as my preferred isotope, with a close third place as cost. When do you usually add internal standards to your experiment? We got a substantial range of responses, but it looks like to the sample dissolved in extraction solvent was the most common. We left that as a multiple response available because many labs will do that at multiple points in their experiment, and I think the results reflect that. Okay, as far as question 7, which should have read, what are the greatest challenges facing the field? It was a two-way tie between unavailability of specific compounds in isotopically labeled form and a need for more commercially available internal standard mixes or kits, which I think is really interesting and something that we should discuss in our discussion today. It's on our list of questions. Great. Well, I hope this was an interesting and informative set of results for you. I am going to leave those results open on my window. If you would so others can view them, I'm not going to stop sharing, but I would just recommend if everyone wants to move them out of the way so our speakers can share their slides. Let's move on without further ado to our panel discussion. And we are going to begin with, so just to review, our panel members will provide a few key perspectives, and they have devised a list of open-ended questions to facilitate discussions at their presentation's end. So in spite of the fact that this is, so we'll leave it up to the individual speakers. If they want to poll the audience right away, then they're welcome to do so. But in general, to keep things moving, I thought we might hold our questions, or even better, put your questions in chat as they occur to you, and we will open things up to a all panelist discussion first before we move into some of our facilitated questions. And I've asked Tim to be monitoring the chat window as we move through. So he will be, and we've got a fairly large group, so when we get to the discussion, I think what we're going to have everyone do is when you've got a question, post the question in chat or say you'd like to make a comment, and Tim will call on you. You can unmute yourself As we get further into discussion, we can adapt that as we need to. All right, so without further ado, let's start. I've asked Lloyd Sumner from University of Missouri to go first.
Right, and you have my slides, and you're going to start. Okay, all right. So our instructions were to have 1 or 2 slides. So I put together a couple of points on what we're doing. I'm kind of going to be very basic as the introductory speaker, but essentially we use internal standards in our lab in both targeted and non-targeted approaches. In our non-targeted approach, I've actually put a listing of the standards that we use. And typically these standards are in our extraction solvents, and they help account for recovery and matrix effects. I'm old, and some of the standards that we use come from the older literature. Specifically, I think I saw Oliver on the line, but many from the Max Planck, so especially for the GCMS. For the LCMS, we use a plant natural product called Embelliferone. It is a plant natural product, so you have to be careful what systems you use it on, so it's not a universal one. But in many of the model systems that we work with, it's just not present at very large or below levels of detection. And we also use long-chain alkanes and or fatty acid methyl esters for retention indices calculations for GCMS as well. So we are using internal standards in our non-targeted approach, and then we use these to assess quality. Next, please. Next animation, Charles. And we use them, again, to assess performance, and this was part of the questionnaire for retention time checks, for intensity checks. But we also use these for normalization of the data post-acquisition to help additional type of alignment of those. Next, please. But those are, we also use external standards for more rigorous type of quantification. And the standard is you generate a calibration curve with the authentic standard for external calibration, and then you get a more rigorous quantitative number. Next, please. But when we want the best precision and accuracy for targeted type of MRM type of analyses, we do use stable label. We prefer C13, but we will use deuterated, assuming that there's no back exchange. But these internal standards, and especially the label, allow for recovery and matrix effects to quantify them in a more precise manner. And then finally, next, please. And then often post-acquisition, we typically don't have to, but if we have a need to cover a wide dynamic range, we will also begin to use weighting factors for our internal label as well. And there's a specific citation there, and that just gives you better precision over a larger dynamic range. Next slide, please. So I just wanted to give you one example of the stable label and type of targeted effects that we use. This is an analysis for kynurenine, which is important in cancer, as well as chronic kidney disease. So typically, next, you can actually just go through all of them. We will generate standard curves, external curves using kynurenine standard, and then we'll add an internal label compound to that mixture as well, and then we actually monitor the ratio of the label to the unlabeled to account for, again, for matrix and recovery effects. And by doing so, we can get very highly precise quantification via this method. And if we have to, and we have had some need to on some of our target analysis, again, where we have a high dynamic range, we will use weighting factors to adjust that calibration curve to give us, again, greater precision and accuracy. So one of my questions was, what are you using? And we'll eventually get to that, but if you want to start putting that information in the chat, that would be nice to know what other compounds people are using for labeling. So that's my 2 slides and 3 to 4 or 5 minutes worth. So.
Perfect. Thank you, Lloyd. Again, yes, we're in a rapid fire format here, so hopefully we're generating thoughts in your minds. Our next speaker will be Christina Jones from NIST, and take it away, Christina.
Okay, hi everyone. I'm actually going to split my time talking a little bit about what we do at NIST within Metabolomics, and then the MQuack Consortium. And so at NIST, we kind of divide our work into two different categories, and it's measurement science and measurement services. And so our measurement science metabolomics work leans to our biological and environmental applications. And so we do use internal standards for both targeted and untargeted studies. And so, for our targeted work, we use internal standards to compensate for any sources of error that's introduced during sample preparation, as well as analysis processes. And so in our untargeted work, we're primarily using internal standards to ensure that our analytical methods are repeatable. We use them to track chromatographic performance, and then to make sure that mass accuracy is maintained as well. For the past few years, I've been more involved in our measurement services for untargeted metabolomics. And so typically, just even beyond omics, that includes calibration standards used for SI traceability and complex matrix standards. And so what's on the slide here is just some of the complex matrix standards that we currently have under development for metabolomics. And so you'll notice that these don't include isotopically labeled internal standards. And so one of the questions I had, and one thing that can be discussed later, is if reference materials either with pre-spiked internal standards could be useful or needed, or whether, more commercial internal standard mixtures are needed for untargeted metabolomics. And so, our reference materials are typically used for long-term QC, well, used as long-term QC samples to assess interlaboratory study data, as well as just inter-study data within your own lab. And so we can go to the next slide. Besides calibration and complex matrix standards, we also work with different communities to develop reference methods and start developing best practice guidelines as well. And so a number of our NIST metabolomic scientists are actively involved in the Metabolomics Quality Assurance and Quality Control Consortium, or MQuack. And so it's just a community-led initiative that was created to develop and promote QA and QC in untargeted metabolomics research. And so MQuack has a few working groups, two of which relate to QA/QC best practices. One of those working groups held an interactive workshop yesterday during MANA, and we can go to the next slide. And so MQuack members who employ LCMS were actually surveyed on their QA/QC practices, as well as their quality management systems. Some of the questions similar to the survey, the poll that everyone just took at the beginning of this workshop. And there were questions, that was related to internal standards. And so I thought this could be a good forum to share some of those responses. This will be published soon. And it was an effort that was led by Annie Evans and Claire O'Donovan. And so there were responses from 23 labs across the globe. They included labs from academia, industry, and government. As you can see, 91% of the respondents reported using internal standards. Most used internal standards to assess chromatographic performance, and mass accuracy, as well as relative standard deviations. They also use them to monitor instrument drift and sample-to-sample reproducibility. The number of internal standards used per method varied greatly from 1 to 800, but 1 to 15 internal standards was the most commonly used. Some labs spiked in the standards into the samples before doing the extraction. Some spiked them into the samples after the extraction. So, this shows that they're being used, but there's not a lot of guidance or best practices on how we should use internal standards, which may be needed moving forward. And so, yes, that's all I wanted to share with my time for now.
Great, thank you, Christina. And again, the speakers have all formulated questions relevant to their topics, and you'll get to see those very shortly. So keep thinking those ideas. All right, so our next panelist is Russ Grant from LabCorp, who's going to tell us about internal standards in clinical and industrial metabolomics type settings.
See my screen? I'm trying to share a screen. Fingers crossed. Did I grab it? Yes. Good conversation. Can you see my screen?
Yes, we can.
And you can hear me?
Yes, we can hear you. You're doing great. Thanks.
Thanks. So what does it mean when you're trying to make a decision for another human? I think it means a lot. As a basic breakdown of the standard LC-MS or GC-MS, you could obviously convert this. A pretty standard workflow, pretty generic. In our experience, internal standard is added directly after a specimen, QCs and calibrators and mixed prior to extraction unless it's determined that the extraction step itself does not affect the relative binding of analyte and internal standard. It is designed to resolve for the majority of the failings of the LC-MS workflow that follow. But in clinical diagnostics, we think about internal standards as a way of designing out the most variable part, and that is the human. In clinical diagnostics and in lab medicine, we're always challenged with the cheeseburger guy that thought it would be a great idea to go and have 4 Big Macs about an hour before they drew their blood, but also putting complicated workflows in the laboratory. So I'm just going to point out some of the things that internal standards provide for us in an environment where the number, precision, and linearity or separation of disease states matter. Here's an example of a 9-step solid phase extraction protocol performed by hand. This is the IS scatter on the left here versus with an automated pipetting system and automated positive pressure manifold. Of course, internal standards we can use to trend across the run, but would you trust a result that was generated with this internal standard recovery down here relative to one that was recovered up here? We actually have a standard in our laboratory where our precision or our total error of internal standard needs to be within 25%. Obviously, that makes that rather impossible because 25% of that scatter is a ginormous number. Okay, so they tell us about the long-term, the downstream effects. What do you think about that chromatographic peak start there? This is homovinylic acid in urine. So it's a pretty robust peak. There's a couple of other isobaric peaks, or at least contributors to the transition. This is MS data. In diagnostics, more often than not, we actually measure 2 separate transitions for every analyte, not full scan product time, but 2 separate transitions using multiple reaction monitoring. And if we look at this peak, the area of these 2 peaks, one divided by the other, that's a qualifying transition. This is a quantifying transition. That area ratio to a neat standard is within 98% agreement. That's pretty good. But if we add the appropriately labeled internal standard, 6 carbon 13s, oxygen 18s, what do you think about this peak now? It clearly is not homovinylic acid. So qualitatively, peak shape, peak retention times, aberration, the ability to actually describe the shape of the analyte peak, even when transition ratio fails, is another wonderfully powerful benefit of internal standards. Appropriately labeled, you'll note. One of the things that we like about mass spectrometry, or in fact external calibration and internal standards that Lloyd pointed to, is it is by definition a reference method procedure. But we should never forget the fact that the interface itself is not an equimolar ionization system. I'll make the point like this. And this is a drug. I understand it's a drug, but I only had 7 days lead time, so these were already approved slides, so forgive me. But this is a wonderful experiment my colleague here at LabCorp, Brian Rappold, showed. So this is gabapentin, and it's D10, 10-deuteron internal standard, spiked into urine. And this is a fast separation, about a minute separation. They co-elute, and the mean of 150 samples of a 1-to-1 mixture of these 2 in previously screened urine is 100%. Slowing that separation down to 5 minutes, we see a hydrogen deuterium exchange problem. This is the unlabeled, and this is the 10-deuteron form, and they don't co-elute. The mean shifts, the variance increases because those 2 particular species are undergoing differential ionization conditions in the source. Moving on. I came from a GLP world in drug development, and so I'm used to running a full calibration system at the start and the end of a batch, and then using the composite of those 2 calibration systems as the overall calibration to actually correct for some batch effect or drift within a batch. But if you're only running a single calibration system or a set of samples at the front of a run or a batch, then you're actually doing historical calibration. And by that, I mean, you are assuming that the internal standard to the analyte addition in the calibration system is consistent through the batch. Now, you could consider that to be appropriate if, for instance, you're preparing those samples in 15 or 20 minutes. You've only really got to generally be concerned about the stability and the precision of your internal standard pipetting for that short amount of time. But in a clinical diagnostic setting, or even in a setting where small numbers of samples hit the laboratory throughout a 24-hour period, nobody really wants to set a small set of calibrators, QCs, and 5 samples and repeat that multiple times during a 24-hour period. The internal standard allows us to do something a little bit special. Here's a design of an experiment. We do this all the time. So we freeze some aliquots distributed in concentration. We store a set, we run some calibrators and the internal standard, and we call that data set comparison day 0. We then store the internal standard under stable conditions. That's solution stability, no absorption in a container, no thermal degradation, no hydrogen deuterium exchange. 24 hours later, or even out to 7 days, and out to 30 days, 30, we take a thawed aliquot of the sample, we add the stored internal standard solution. We now have to add them precisely volumetrically relative to day 0. So there's no calibration when we run the next day to validate this. And then you're simply solving for the analyte to internal standard peak area ratio using the regression equation that Lloyd just showed you as an example. So when you compare day 0 calibrators, samples, and internal standard added to, in this instance, 1 day later, using a thawed sample, so both the day 0 and day 1 samples go through the same freeze-thaw cycle, so there's no freeze-thaw variability in the actual assay design. Stable internal standard stored under known stable conditions, and you compare yesterday's calibration with today's materials. Again, this is drugs. It also works for metabolites. I only had 7 days to put these slides together. And there is a lot more one can do, because if you think about it, a reference method procedure as mass spectrometry can be and is, the value of internal standard to correct for the sins of mass spectrometry, but again, also using precisely prepared samples from day to day to day allows one to use historical calibration. And I will come back to the first thing. The biggest variable we have to deal with in the clinical laboratory is both the people on every day, but also the patient specimens we get to analyze. And so we use this with automation protocols that are precise from day to day. Humans are the biggest variable, quod erat demonstrandum.
Great, thank you so much, Russ. Let's move along. I'll see if I can reshare my screen. And where is my share screen? Let's jump back down. All right, our next speaker is Matt Merritt at, from University of Florida, who's going to speak to us a little bit about internal standards in NMR metabolomics. Matt, take it away.
So this is the only time I get to have a great deal of pride in doing NMR. Is we have well-known abilities to do quantitative metabolomics in a relatively straightforward manner. The only thing that you really need to do to collect qualitative data is make sure that the time to repeat for your NMR experiment is known and the relaxation times of your compounds is known. If that's the case, then the relative peak areas, once you normalize for the number of hydrogens you're looking at, tells you the concentration in a remarkably accurate way. And it's been shown that this good quality of MR scales across laboratories in a very straightforward way. And so that's something that for targeted metabolomics by NMR, if you've got a certain small set of compounds, 10 to 20, that you want to know, then NMR is going to do a really great job for that targeted metabolomics. As far as internal standards, primarily, we always think about a chemical shift standard. That's been known since they started doing NMR on electromagnets that drifted all over the place. And they said, okay, let's put a chemical shift standard into our mixture or whatever we're running. And so tetramethylsilane, obviously, you remember from organic chemistry. For metabolomics, you see DSS, TSP are very commonly used, and they all resonate at a frequency that's far away from any metabolites. So one of the central issues that remains is, however, for us is pH effects. And so that's going to come into play most commonly in trying to do metabolomics in urine because if you're going to do it from serum plasma or something like that, normally you're going to do an extraction to get rid of proteins and stuff like that. In that case, you handle your sample in such a way that it comes down at a pH that you know and understand. But if you're doing a urine, you've got an option, and that is buffer the urine till it comes to a standard pH, and then just have everything relative to your chemical shift standard. Or the problem with that is knowing how much buffering capacity you actually need. How far out are the urines that you're actually taking a look at? And so there's been quite a number of searches for metabolites that have a good pH dependence. So creatinine is one that's commonly occurring, but it's got a chemical shift difference that is only really very effective from 3 to 5 ppm. 3 to 5 pH units, not closer to normal physiological pH. And so we do use a variety of different chemical shift standards that are pH dependent. If you go to a molecular imaging conference, you'll see about 50% of the talks will be about pH sensitive compounds, however you're measuring them. So I won't try to elucidate all the different compounds or name all the compounds that you could use. The internal standards for spiking and confirmation of your ID is a little bit different for us versus LCMS or something with a lower detection limit, because we actually probably need a fair amount of compound, and that's certainly something that we have to take into account if we ever want to order a standard. But we don't normally need isotopically labeled standards. If we can get the pure compound and put it in, that's going to be fine. I see Lloyd asked the question, how important is pH monitoring in non-aqueous solutions? If you do like a Folger extraction and you want to do metabolomics or lipidomics, then you'll find, to my experience, that pH effects are probably negligible. You don't have to worry about that too much. Okay.
Great, thank you, Matthew. Let's move on. As I mentioned, we invited Alla to talk, and unfortunately, I didn't give her quite enough lead time to get slides approved. But I wanted to open up, Alla, before we move into our open discussion. We had a great talk the other day. Did you want to add any comments relative to internal standards in pharma and industry to our conversation before we move forwards?
Yes, thank you, Charles. Sorry, I cannot show any slides, but I can speak to our general practices as they are relevant for the industrial setting. And in our work, my group is focused on biomarker discovery and analysis. And we are supporting projects at different phases in the pipeline, starting from discovery, ending up in clinic. And we have to use a combination of global and targeted profiling approaches. And I will start from the initial phases, from the start of the project, and going through the phases of the pipeline, because we use different methodology and reach. So for discovery, at the initial phases, we are working with typically different types of materials, cells, tissues, biological fluids. My group members are joking that they used to be analytical chemists, now they're just collecting body parts. And this is because we really had to expand type of human materials that we need to profile. And the reason why I'm mentioning this is because, as you can imagine, you switch the material, you need to do a lot of optimization. So standards are very important for that purpose. So the second phase is developing a screen. And typically screen is based on the targeted method. It is biochemical screen where we monitor single biochemical reaction, but we prefer to do cell-based phenotypic screen where we have closer to real life case scenario. And as you can imagine, the matrix is complex, but we are typically monitoring 2-3 biomolecules with corresponding internal standards, because at this point, our screening methods are LCMS based, but relatively high throughput, up to 2,000 injections per system per day. So we need to use isotopically labeled standards. Once we select active compounds, we have to follow up with the fingerprint, because we want to be selective, we want to understand mechanism of action. And typically what we do knockdown experiment in cells, get desired fingerprint for ideal case scenario, 100% selective inhibition. And then we also may do a couple of fingerprints for undesired target that we don't want to hit. And then we profile each compound that looks like it has good potential and try to match fingerprint. And at this point, typically we run each compound at a couple of concentrations below IC50, above IC50, and this response has to be quantitative and consistent. So we really need to be at least semi-quantitative for the desired compounds, but this is already a global approach. So our global method has to be optimized to give us a semi-quantitative approach. Then once we select promising compounds, we move into animal trials. And typically here we monitor biological fluids. We also look at tissues, and we use combination of targeted and global profiling approaches. And then after that, in clinical phase, we also follow up with targeted and global approach combination. So while with targeted approach is more or less easy, we use relevant internal standards. Unless they are not available, if we cannot use them, then we are forced to use matrix matching or standard addition methodology, or in some rare cases, we can use external calibration curves. For global approach, we really need to understand what we are measuring, what we are able to measure semi-quantitatively, and adjust our methods based on global approach. So we have developed internally a mixture of 63 standards. They represent different metabolic pathways, different classes of molecules, and typically in our methods, they elute all across our chromatographic profile. So with this standard mixture, we typically can develop a method that either has a good global coverage, or can focus on a particular class of metabolites. And then, of course, we work with different tissues and we verify semi-quantitative approach. We have come up with the protocol how we do that for different biological materials. And then once we are happy with the method, we typically use only 5 in some cases 10 spiked isotopically labeled standards, just predominantly to monitor our consistency across the chromatogram for retention times and instrument performance. But we use some other controls like total metabolic average that we monitor and use for normalization. We use pooled sample as our system suitability, and we run it every 10-15 injections. And also we run our standard mix periodically along with the blank. And for global method monitoring, this really is sufficient for us. In terms there is one other application when we want to zoom on a particular metabolic pathway, and especially in the situations when we want to monitor de novo synthesis of metabolite, we need to use standard isotope labeled material to follow up with metabolic phase of a particular precursor. And for that, we need large quantities of specific metabolites, which brings availability issue because it's expensive and availability is not where it should be to our mind. I think I kind of covered the scope of our work, and in terms of the questions or comments, of course, I would like to see a collection of standards that would allow us to look closely at a particular pathway or a type of metabolites, like focused collections that would be available from the vendor for us. And one idea I would have to make this available, and maybe this is a good discussion point, is it looks like each vendor that generates isotopically labeled metabolites that we could use, they are working independently, they don't have connection, so we can buy some materials from multiple vendors, but in terms of variety, this is really not where it should be. So maybe in addition to consortium of scientists which are using isotopically labeled materials, we could bring to the table vendors, so maybe they can split the spheres of influence and broaden the scope of materials that we can work with. Thank you.
Thank you, Alla. And I think that last point is a valid one. And I think we have vendors here today eager to hear what we have to say and to contribute to discussion. So we have somewhat limited time, but we really do want to hear your voice. So this is where we get into the discussion. We have a lot of discussion questions. There's no way we're getting through all of them, and that's fine. But basically what we're looking for here is for your input on what we as a community can do better based on what you just heard. If we could, let's try out putting questions into chat window when you have something to respond, and Tim will bring up the comment and unmute you, or you can unmute yourself. If we move forwards, we can, we'll adapt as we need to. Okay, we'll look at some of the questions from all of the panelists and some general questions that we came up with. But the first questions on the list are pretty basic ones in terms of what internal standards you use and why, and then we can fold in, do you use them for targeted and non-targeted metabolomics? We already saw some of those results on the survey, but does anyone have specific comments to those questions as to how you pick your internal standards and how we all should be?
So this is Russ. I would just add a comment relative to the fourth one. There are very, very few software systems that actually do anything materially valuable in internal standard monitoring in general to make that tactile for the analytical scientist or the data reviewer. A massive gap in opportunity, and by tactile I don't mean looking down rows and columns for a number that's shifted left or right. I mean literally flagged for you. We specifically use a technology made by Indigo Bioautomation that does that, and they don't pay me anything to say that. Although Randy Julian is a very good friend of mine and is in my band.
Can you document that in the comments, Russ, the Indigo software, and maybe a URL link or something?
Yeah, I can. Will do.
Other comments? Tim, do you want to, do you have comments from chat that have popped
There
up?
are, if there's some, and I can, we had one come in about from Yassine. I don't know if Yassine wants to unmute and ask their question.
Sure.
That's you?
Nobody really talked, people talked about the general workflow, but not very specific matrices, right? So while I assume that, for example, LabCorp might work a lot on urine and plasma, and Lloyd Sumner might work more on plants, there are others who have interest in mouse organs, for example. So mouse organs have different concentrations, different compounds. The brain is different to a heart and whatnot, right? So how do we get kits of internal standards that have the right concentration for the right compounds and not just blood?
Yeah, no, so can I just address that quickly? If it wasn't clear in my workflow, the first part of the very first step of any analytical experiment, be it blood, tissue, mouse, goat, horse, or the cheeseburger guy, is in fact generating a homogeneous specimen and generating a miscible mixed, in our experience, and we do tissue work as well, a miscible mixed internal standard to analyte composition, miscible prior to extraction. And in fact, any combinatorial problem like a massively multiplex metabolomic experiment, I guess the question you're asking is, can we get a cocktail of very concentrated that one could then potentially dilute to the appropriate circulating range in your samples? But I think of that like a tray of round fruit, and you're always trying to keep the orange from falling off. There's just too many variables in multiple metabolomic space. But I get the concept. The thing that I wanted to get across was the mixing miscibility and heterogeneity in the use of internal standards, as much as you, I think you're talking to, Oliver, a composite of internal standards in the appropriate response or concentration space specific to tissues and different animal species.
Yes, and also not only that, but also some kind of a consensus, or at least some kind of discussion about how to move the community forward in terms of which compounds are important for which organs, you know, let alone the concentration.
So if I could take a stab at a response, Oliver. You know, David Wishart and their group and the HMDB keeps, they do the relative quantitation or absolute quantitation. And I know TMEC is now beginning to consider the production of kits. And I think that these kits could be tailored towards specific applications and concentration ranges, which I agree can be a little bit of a challenge to figure out, you know, or at least it takes time to figure out on your own. So I would encourage you to visit TMEC and see what they're doing these days, or help push them forward. Are you planning any kits yourself? Chris says.
No, I don't think that any academic lab should make kits. I think this is myself, I think this is the obligation or the interaction of the metabolomics community with corporate vendors. Who can guarantee the stability and availability of kits over time. But that, you know, that's my private opinion, I guess.
Well, TMEC is kind of a hybrid between academia and you know, commercial type of entity. You've got a few spin-offs, but I thought Beecher was going to say something too.
I was actually, Lloyd, I was sort of impressed by one of your slides in which it appeared that you had a constant amount of the internal standard and you had a range of, you were using it to measure, and it appeared fairly accurate. I didn't get to study the slide very long, but it appeared that you were accurately measuring over a range of, maybe 1 to 20 down and 1 to 20 up.
Yeah, I think the Kinnearing, the dynamic range there was about 1,000 or more, a couple of thousand.
And so Oliver, I mean, I understand exactly what you're saying. And I understand that if you can get the internal standard at exactly the average concentration, then you will have the maximal sensitivity for quantification. No two ways about it. However, I think that dynamic range that Lloyd showed, really suggests that if you can get an internal standard in there for metabolomic work where you're trying to get, a broad spectrum, if you can get an internal standard in there that is, even remotely within range, you can get enough utility out of it that you'll actually get a reasonable result. And I'll leave it there. And obviously we make and we completely agree that internal standards are the way to go. And we believe that, biochemically complex internal standards are the least expensive way and therefore likely to be the most, opportunistic way to move forward, for metabolomics itself. It's just, if you have to buy these things chemically pure and then remix them, it's like, washing a glass and then drying it before you fill it with water. Why bother? It's just, it just seems that if we can make mixtures that are pretty close and use them as internal standards, that dynamic range suggests that we can get away with much better data than we're currently getting.
There's a discussion about deuterium versus carbon labeling in the chat, and I don't know if we want to raise that as one of the comments to talk about, since we have a number of experts that are in the field for this.
Yeah, let me just step in, and I think Russ illustrated quite elegantly the point that, the chromatography for deuterated compounds is never the same, and it's always a potential flaw. Back when we were doing GC, by the time you got out, past about 15 minutes, the two peaks were the deuterated peaks were no longer tracking appropriately, and so there was a great deal of inaccuracy coming just because of deuterium. I really have personally never liked deuterium. It's cheap, which is why it's used.
I also have something to add to this point. In the past, I absolutely resisted working with deuterium labeled materials, but unfortunately, carbon-13 is not always available, carbon-13 version, and also very expensive, especially when you start using them in animal trials. So unfortunately, in industrial setting, we had to do what we have to do to move forward. And if carbon-13 material is not an option, then we are forced to work with deuterium. But especially in global setting, the value that labeled material brings is really stability of performance, because we are semi-quantitative. We need to make sure that we detect the difference. And we do optimize our methods specifically for that. And to that regard, we are no longer running a standard metabolic method, which has the same gradient, the same extraction procedure. We are changing it so quickly for each tissue type, for each animal model. We have maybe not so unrelated versions of the method, but still different. And of course, it's nice to have a representative matrix commercially available, but for each animal model, for each type of experiment, we typically ask our colleagues in vivo biology to generate matrix for us, and we spike our standard isotopically labeled material into this representative matrix, and look at our method performance in that specific matrix. I'm not sure how universal we can make, a matrix spiked, and how applicable it will be for a variety of experiments. There is value, of course, but there are pitfalls also.
Great.
Charles, I thought these were rather rhetorical questions. Maybe they answer themselves, but do you trust an LC-MS result with everything that we know about the electrospray source or the APCI source and all the weird biology going on to measure or quantify a result to make a decision and inform another human being of a life change? Would you do that without external calibration and internal standardization? Or another way of putting it is, have you ever seen published a representation of fold changes with analog internal standards using infusion, FIA, LC-MS, whatever, that actually are consistent with fold or concentration range changes that you see using the reference method process? And I do acyl carnitines and organic acids and amino acids and free fatty acids by GCMS and LC-MS. We do all of that work as well, and there is not internal standards for all of that. I just wanted to throw, I think these are kind of self-answering because I've never seen the second one done right.
Well, and you also have discovery research versus,
Clinic.
high-level quantitation. And so, maybe these are workflows, internal standards in a discovery platform might be, and is slightly different to maybe Ala, there was a question about her having 63 standards, which seems to be probably on the high end of anybody adding standards for a non-quantifiable or an untargeted approach versus say 1 or 2 that's added. That's a pretty big range just for measuring compounds, keeping in mind that we measure, thousands of metabolites in a single untargeted method. And so there, the validation and validity of some of that is something that we should be discussing. And Russ, those are key comments, and translating information from one place.
I only make the point that ion suppression is in a mass spectrometry paper in 1950, and the first significant electrospray paper mechanistically from Kabali and Tang in 1994 also demonstrated differences in ionization efficiency as a function of structure that you can truly only resolve with a biochemical mimic that is appropriately labeled, and that's really the point. Or another way of saying it is, good luck on the biology and the discovery, because it's a hard yard. It's hard to go from left-hand side of that equation to the other side.
Yeah. Well, I totally agree with Tim in that the discovery platform and the quantitative platform, or the clinical testing of biomarkers, are two separate workflows. And the discovery, the things that we do discover, typically are greater differentials. The resolution of the discovery experiment is limited. So if we're looking for 5% differences, then that's almost an impossibility because of the biological variance alone. But once we do have a reasonable biomarker, I agree that it's critical that if we're going to do targeted clinical work on that we need the internal standards to account for recovery, suppression, and all of those and above. But again, I concur that it's two separate workflows, and we work on both of those ends, in that we try to do the discovery, and then ultimately, if there's a clear need for the clinical high rigorous quantitation, then we move to more targeted type of methods.
Yes, I really agree with that. And in global profiling, which is predominantly the discovery part for us, if we don't have quantitative response, most likely we just will not see the change. We will miss a biomarker. And our goal is, especially like with the standard mixture, we use it only in method development phase and then as a system suite. So we just make sure that we are covering in semi-quantitative fashion the pathway or target or group of metabolites of target, or just as many as we can. And we have a digital tool that allows us to do that. And we are preparing a publication on our approach. But with quantitative assay, which is a follow-up, and this is a confirmation that you do have a true biomarker, then there is no way around it. We need an internal standard.
Great discussion. I would on this, so we're drawing to a close to a close, and I think we've had some great discussion. I would, on this slide, I would draw everyone's attention, and Christina can comment. I'm particularly interested in question 2, in that we've talked a lot about targeted, and I think there's general consensus that in a targeted assay, you want an internal standard for every metabolite that you're trying to measure if it's possible, and although it sometimes isn't, but untargeted is kind of the big open question here, right? No one has a consistent way of dealing with internal standards of in untargeted experiments, and how many is the right number? How many is enough? How many is too many? Do you just try to span your attention time range decently? Do you want, do you try to make, to do as good quantitation as you can out of the untargeted? What's the right answer? I'm interested in short term, what's the answer to the number of internal standards, the classes of internal standards for untargeted?
Charles, if I can throw in my 2 cents here, because we actually have spent a great deal of time on exactly this. We use as complex a mixture as possible in our case.
Or using our very efficient gamma irradiation.
So, and in addition, because we've got so many, we end up doing a quantitation on a Mustis algorithm, that is the total area under the curve for the 2 samples, and we then normalize the C12 sample, the natural abundance sample, to the C13 internal side. The C13 internal side is kept at a constant, and so therefore we are always normalizing to a constant. That normalization is actually applicable to every compound in the sample, and so we're able to normalize those compounds that do not have an internal standard. And I think that's the part that I was trying to get to for you, because I think you can run an unbiased analysis in using a sufficiently complex internal standard and actually assist and better the results on your untargeted side.
Thanks, Chris. Any other comments here on this slide?
Charles, I just think there's this philosophy of chromatographic separations versus an infusion or a flow injection experiment, and whether a flow injection bolus can be corrected in a manner more appropriate than a chromatographic separation. One of the things that I've seen presented by Gary Van Berkel in a very different context is infusing a constant flow of a combination of internal standards whilst doing something like a chromatographic separation. So you have a constant infusion versus a chromatographic peak. And unfortunately, we don't have the tools to do normalization with a constant infusion as the denominator. We do peak over peak, not constant infusion. So we still have a software gap on it, at least creating some degree of hybridization of infusion normalization with a handful of internal standards versus chromatographic normalization, where generally you just can't put enough of them in there if you want really great data.
It's an interesting approach. Well, we are reaching the end of our session here, which is scheduled to end at 3:05. So I am very sorry to cut discussion short. We had a lot of other great questions. I think we had a great set of panelists. I think to me, we've highlighted that there is a diversity of opinions on this issue of internal standards. What I'll say is that I hope to compile the notes from this session and distribute it out, and maybe maintain a dialogue. And what I would encourage all of you to do, if you had thoughts that didn't get to be shared, is to engage with long-term working groups that are interested in this. That could be MQuack, and it could be the NIH Metabolomics Consortium through Tim or I or others. And I think a key point is that vendors are eager, from my experience, to hear our needs and our ideas, and they should be a partner in this process. And so, although we didn't have enough time to get into to solve the issues of the field today, we should all be thinking about how we can help make these ideas, make interlaboratory cooperation on these issues and vendor cooperation a reality. Please, let's stay engaged on this topic. And I look forward to continuing to correspond with some of you in the future. So thank you very much for participating in this session.
Thank you, Charles, for organizing. Thank you, Tim, for your efforts, and my fellow speakers. Charles, could you somehow capture the chat text? Because I know there's been a couple of references posted in there. I've tried to copy it, but I can't do it. So I don't know if it's
I will capture the chat. I believe I have it set to save, but I'll make sure I do before the meeting ends.
Okay. And just email it. That'd be great. So appreciate it. Take care, all.
Thank you very much. It was a great discussion.
Yeah, it was. Hopefully we can stay engaged. Thanks, Charles and Tim.
Is the chat staying open for just a minute? I'm going to try and grab that link that you're asking me for.
Sure, we'll leave the chat open for the next 10 minutes or so.
Just give me a second. I think Gary Van Berkel has presented that. Thank you very much for the invite.
I think I remember that as well.
It's the work he was doing with the tissue microdissection, and he was looking at propranolol, I think, and then doing a constant infusion of propranolol internal standard. And they had to develop their own internal technology to actually do an integration with an infusion as well as a chromatographic overlay. We also worked with Randy Julian on that. We've never moved that one forward, so whatever.
Yeah, there was another individual who did something with an acetonitrile proton bound dimer, which actually reflected some of that work too. And I'll have to look at that reference, because there was some interesting work about using that dimer as a measure of electrospray stability and normalization.
Oh, that would be Rick King.
King, yeah.
Yeah, that's, and he actually patented it. I'm trying to find.
He did.
Yeah, so we tried that as well. And we tried, and you can get basically trying to use clusters to infer variations in ionization response. It made no sense. We spent about a year on it, and I still don't understand it. Made no sense to me. It's taken me a while to. What was the session you came from anyway, Tim? Hardest working man in show business you are, by the way.
Not anymore. I came from metabolite identification session that I was chairing.
I found the reference. It was analytical chemistry in that long ago, a decade ago. Wow. Give me a second. Let me just. This is at least, I think, the first one. I remember seeing this at ASMS. So, God, I wish there was a nice way of doing copy paste. This looks a mess. I apologize.
Got it. All right.
So I see Felice is on, Chris isn't. So I understand that you're doing derivatization of two separate specimens in a combination. Is that right? Or are they combined in assay? I think, Tim, I know you've used the Eroa workflow, right?
Yes,
Can you recombine? Independent derivatization and recombination.
What is that?
You split the sample into two and you use two separate derivatization strategies to generate the two different C12, C13 patterns. You recombine them and assay them as a single specimen. Is that correct?
I don't know I don't know the new one. The old one was just a isotonically labeled yeast that was grown and isolated and used as a reconstitution standard. That's one we use. I don't know about the new one.
Well, yeah, the workflow is, anyway, hi Russ, hi, hi Tim. The workflow is such that we have, it is a fully labeled yeast standard, and we have two samples. One is the 95% internal standard that is spiked into the experimental samples, and the other one is a 95% yeast extract mix 1-to-1, which is analyzed every 10 or so samples. That way we could build a reference library and also use that, we call that the long-term reference standard. And we've measured the stability over 5 years, and it's perfectly stable. But it tells you a lot about your platform stability. Of course, you look at your
now I understand.
Yeah.
Yeah, now I understand. But it doesn't tell you about differential ionization efficiency of these unknown metabolites. And I keep coming back to this: the electrospray process is not a universal ionization system.
Well, we use it to, as Chris mentioned, we use it to normalize, but we also use it because it has 800 compounds or more. We use it to correct for ion suppression.
You mean those yeast materials are added into every patient specimen or every study sample, or are they still just every sample? They're added in. Well, then again, I reiterate those, unless it is a mimic of the analyte of choice, it isn't going to behave the same way. We know this from the service activity work from Chris Enke. You get differential ionization efficiencies. It's the cheeseburger guy. Blame the cheeseburger guy. That's what I'm saying.
You mean if it's not the same metabolite?
No, he's meaning the matrix changes in a clinical setting from someone who has a high fat diet versus someone who doesn't. And that alters dramatically. That's the cheeseburger effect.
Well, and then kidney disease. I mean, we have to come in with the central premise that the person is not normal. And in fact, I've only ever seen one normal human. He was in California somewhere. I think he was a hippie. He was very, very happy. So what's normal, right? I mean, we have this incredible tool, and I don't begrudge you the challenge of trying to measure so many things with confidence. I think all of these tools add value to the question of confidence.
Yeah, no, I mean, we've had some great, we've done it with, lots of different matrices, and we haven't seen any issues. We're going to, we're working on a round robin to look at, across labs, interlaboratory. We've done it with everything from cannabis, to blood to urine, and we just don't see these matrices. This matrix effect.
That's not possible. Unless you're using GCEI with TMS derivatization, that is not possible.
Well, maybe we can, show you some data and take it offline, but, Chris has got some great data.
It's the Achilles heel of the electrospray process.
Well, happy to, as I say, we just don't see the matrix effect. But I'd like to, continue that discussion.
Well, Tim, Charles, thanks for the late call up. I appreciate the request. And are you going to share the slides to the people who signed in?
I'm going to just ping all the presenters one more time to make sure that's all right. But my plan is to share the drive, we'll find out what they have planned as far as MANA, as far as slide distribution. But I don't see why not to make the slides available to the whole MANA consortium, really, through the MANA website. So that would be my plan.
Mine are cleared, so you can share them. Thank you, guys. I really appreciate the
Perfect.
time. And,
Well, thank you so much. Your slides were great, and I think we did have a good discussion. I think I learned that we're all in, as somewhat predicted, we're all in a little bit different places as far as what we think the greatest needs are, and how we do develop our protocols. And it really, if nothing else, it confirms that to develop. It isn't possible to create a totally one-size-fits-all solution to internal standards or references, but to the extent that we can work together on this, we have a long way to go, both on the adoption of best practices, right? I think that's one thing that may be a little bit easier, but then as far as really standardizing procedures and metabolomics data that comes out of it, so that an analytical lab has a chance of getting the same quantitative results as would come out of a clinical lab. I think we have a lot of work to do ahead of us, and I think it's it's informative and kind of fun to see that through these types of discussions.
Yes, indeed. Nice job, gentlemen, and nice job, all the speakers. Thanks again, Tim. Give me more than a 7 days heads up next time.
Yes, I will, and I apologize. I was late in getting speakers out, and you were probably one of the very latest that I contacted, so I apologize for that. But thank you for being a good sport about it, nevertheless.
Of course. Thanks, guys, and thanks.
All right, thank you. All right, I'm going to end the meeting for all. Have a good afternoon. Enjoy the rest of the conference.